things now. Okay, excellent. The sound quality actually visually is pretty good. So we're going to start talking about microbiology. I'm going to introduce you to this science in general. Okay. So what are the subjects of microbiology? All things small. Okay. We call them microorganisms. Microorganisms, majority of them can be seen in the light microscope. So you have these guys can be seen in the light microscope. That will be bacteria, can be seen in the light microscope. Unicellular eukaryotic organisms can be seen in the light microscope. Viruses, in general, cannot be seen using regular light microscope like the one that sits in the cabinet or the one that sits here on the desk. To visualize viruses, you need electron microscopy. Okay, so size-wise, viruses are going to be smallest. Then, generally speaking, you're going to have bacteria. Then, generally speaking, you're going to have single-celled eukaryotes. And then, the largest organisms that are studied by microbiologists are helminths. Helminths are worms. Okay, so they're large, they macroscopic. So they can be seen with a naked eye. So far, we're good? Other exceptions from this general rules? Yes, of course there are. There are viruses that large enough to see them using that microscope. They fairly rare, but they exist. There are super small bacteria that you need electron microscope to really see. And there are bacterial cells that are so large that they can be seen with the naked eye. They still kind of tiny, but you can see them with the naked eye. But we here not focusing on the exceptions, although they're fun. We're here focusing on the general principles. The general principle is that viruses, electron microscopy, bacteria, single-cell eukaryotes, light microscopy. Helminths are large enough to just see them. Now, all these organisms can be categorized based on the cellular structure and number of cells that each organism is made up. So these two categories, the bacteria and archaea, and we're going to talk about the difference between these two. You can see the exemplary bacteria here. It's cells of Yersinia pestis, causative agent of the Black Plague. Bacteria and archaea are both prokaryotic. They do not have nucleus, and they do not have any membrane-bound organelles. Interestingly enough, all prokaryotic organisms are unicellular, no exceptions. The modern biology, modern science, doesn't know about any prokaryotic organisms that would have more than one cell. So when you look at bacterial specimen and you see a lot of individual cells, you have to understand that each cell is an organism. They do cooperate, but they cooperate as organisms. They don't develop tissues like higher animals do. And bacteria are studied and archaea studied by bacteriology, kind of obvious. Algae. There can be big macroscopic algae, multicellular algae. And there are unicellular algae. So they can exist in either sort of form, right? So you go to the lake, you go to the ocean, and you can see with your own eyes multicellular algae. Now, single-celled algae is great, probably the best example of those. Sometimes you can see the bodies of water like ponds and lakes having this green growth in them. That's unicellular algae. This, for instance, this one, love this. It's Emiliana huxleyi, gorgeous unicellular algae that can be found mostly in the tropical waters in the Pacific Ocean. To give an idea how important this just one species of algae is, Emiliana huxleyi these cells produce more oxygen than all forests in the world. They photosynthetic organisms. Now, protozoa, this one here is a typical protozoan cell. It's the pathogenic single-celled microorganism called Giardia lamblia. Protozoa, by definition, are unicellular eukaryotes. Makes sense, right? So they're unicellular eukaryotes. They always unicellular, no exceptions. They are subject of study by protozoology or parasitology. Speaking of which, I failed to mention that algae are studied by phycology. Phyco. Phyco means algae. Now, fungi. This fungus here is called Candida albicans. You may not know the Latin name, but you know very well the disease that Candida albicans causes. Generally called candidiasis, but you may know it as thrush. Okay? So, Candida albicans is one example of unicellular fungi. But fungus can grow as multicellular organism. Molds that you can see on the surface of the bread, or berries, are the example of multicellular fungus. Fungi, as other organisms that we're talking about now, are eukaryotic, and they are studied by the part of microbiology called mycology. Okay? Now, helminths. These organisms hold dear place in my heart, not literally. My third favorite organism after my dog and the viruses. I probably should include my wife somewhere there. Helminths are multicellular. They're large. So this one, Tinea saginata, or beef tapeworm, for instance, can grow as long as 30, 40 feet. Okay, so they're gigantic. Now, they are studied by helminthology or parasitology. And finally, viruses. Viruses are acellular, meaning they do not consist of cells. They are practically the assemblies of proteins and nucleic acids that can reproduce in other cells. That's important. Viruses, all viruses, are obligate intracellular parasites. They require a cell to reproduce. Okay? They are kind of like a seed of a plant. If you have plant seed, it cannot divide, right? But if you put it in the soil and it starts to grow, it becomes a plant, and this plant can produce more seeds. We're good? Obviously, viruses are studied by virologists. So this is an Ebola virus. I'm going to talk about it at some point. But I want to summarize what you need to know from here. I will ask you, fungi are studied by mycology. Make sense? Bacteria are unicellular prokaryotes. Protozoa are unicellular eukaryotes. We're good so far? So you have to understand whether certain type of microorganisms are eukaryotic or prokaryotic and how many cells they have. Okay? Now, one really interesting question that often arises is why prokaryotes do not form multicellular forms. Why they do not produce organisms with tissues, like differentiated tissues. And the basic answer is we don't know. There is no satisfactory explanation of why this does not happen. Most likely, and this is sort of an accepted hypothesis, is that bacterial or archaeal cells cannot establish a sufficient level of communication between the cells. That makes sense here. Think about this. In higher organisms, say humans, all cells very closely communicate with each other. They establish short distance, autocrine, paracrine communication. They establish long distance communication like endocrine system. They actively secrete, they actively transport chemicals in, and they all differentiated. Probably bacteria cannot communicate intensely enough to develop differentiated tissues. Are we good? Any questions? Now, before we actually start talking about microorganisms, we need to talk about how we classify them. First classification was developed in 18th century by Carl Linnaeus, who was a botanist, Swedish botanist, and he wanted to classify plants. And he developed what we call a binomial nomenclature. So when I say here that microbial names consist of two words, like Giardia lamblia or Bacillus anthracis, you see here, not only microbial names, how do we call humans biologically? Species. Homo sapiens. Homo sapiens. Homo is the genus name. The word sapiens defines the species. Now, I want to make it very, very clear. It's kind of weird, but if we talk about any organisms, any living organisms that carry binomial name, like Homo sapiens, or Felis catus, which is house cat, okay, or Lupus lupus, which is gray wolf, or Giardia lamblia. First word always means genus. In the hierarchy of groups on the right, this multicolored hierarchy, you see genus is right here. Genus, all different species in the genus are very, very close. Say you have a genus of Panthera. Panthera leo, Panthera pardus, Panthera tigris stand for lion, leopard, and tiger. They're all very similar animals. They all have fairly similar ecological niches. They all have fairly similar behaviors and stuff like that. Does that make sense? They're all different species belonging to the same genus. Since they all belong to the same genus, they all bear the same first name, Panthera. So if you would look at microorganisms, say genus Bacillus, you're going to have Bacillus subtilis, Bacillus anthracis, Bacillus cereus, Bacillus megatherium. They're all very similar species, but different species. That makes sense here. So, if I ask you, what does the first word in the binomial name stands for? You tell me. Genus. But how do we call species? Two words. So, say Bacillus anthracis is species Bacillus anthracis of a genus Bacillus. Giardia lamblia is species from the genus Giardia. We're good? We got binomial nomenclature. Viruses are different. Virologists are kind of outcasts because they deal with acellular organisms, which makes viruses not living. We'll talk about it. But viruses are named weirdly. Say, there's a virus known as influenza, right? It's called this way somewhat because of the disease that it causes. And then you may have heard about viruses called MERS, Middle Eastern Respiratory Syndrome, called for both where it was discovered, Middle East, and the type of disease that it causes, respiratory syndrome. Ebola virus is named after the, I think it's village, oh no, it's river Ebola. In the whereabouts of Ebola River it was discovered. So, you know, mainly the general rule for viruses, people try to name them after the place the virus was discovered. Sometimes it creates some very unpleasant consequences. You may have heard about norovirus. Some people mistakenly call it stomach flu. It's not a flu. Winter vomiting bug. Two-bucket disease. All those wonderful names. Norovirus was discovered and first isolated in the city of Norwalk, an hour west from here, in Ohio. So, and it was called Norwalk virus first. And, you know, which city wants to associate its name with a virus that causes diarrhea and vomiting? So, people from the city actually asked to change the name, and the name was changed to norovirus. So, you know, kind of can be weird. Now, classification. The modern system was established, one of the modern systems, was established by Robert Whittaker in 1969. Whittaker proposed a system of 5 kingdoms. Monera, that would include both bacteria and archaea, and we'll talk about what the difference between those two. So Monera would refer to all prokaryotes. Okay. Protists, which would refer to all unicellular eukaryotes. And then plants, well, kind of obvious. Animals, also obvious. And fungi, also obvious. Does that make sense? Now, Whittaker classification was based mainly on the observable characteristics. Say, structural. Look, you have all prokaryotes, all cells that have no nucleus. They're all unicellular organisms. They're all rather small. They're all similar. It's just logical to put them all in the same kingdom. That make sense? Clear? Okay. Now, you look at all the plants. They all share similar, structures of the cell wall, types of cells, mesophyll cells, They all share similar tissues and whatnot. Okay, we put all the plants in the same kingdom, right? You look at animals, you look at tiger and the leopard, they're very similar. We're going to put them in the same genus, okay? You look at, dolphins, all dolphins have lungs, they're all mammals, we're going to put them all together, right? So that's Whittaker classification. It was based on ecological characteristics, anatomical characteristics, observable things. And then in the 70s, a gentleman named Carl Woese, unless I tell you to memorize the name, you don't have to. Carl Woese was studying bacteria. And it was 70s, so a lot of molecular methods were already in place. So his idea was pretty simple. You're all familiar with the concept of genome and genes, right? So what Woese was doing, he was specifically identifying the exact sequence of one gene, 16S RNA gene from bacteria. It's a gene that is present in all bacterial species, all of them, no exceptions. And this 16S RNA gene encodes, surprisingly, an RNA that is a critical part of a structure called ribosome, necessary for protein synthesis. Are you with me so far? Now, you can imagine, protein synthesis is super important, right? So if something is necessary for protein synthesis, this gene should be really, really conserved. Do you understand the word conserved in this case? Conserved in biology means doesn't change easy. So say, if we would sequence genomes of all people in this room, and we will compare them, there are going to be differences. Makes sense, right? But if we would take genes that are critical, say RNA genes for ribosome, and compare them between all of us, I bet there aren't going to be any differences. They're going to be absolutely identical. They're so important that a minor change may screw up the entire protein synthesis system in a human. Does that make sense? So Woese's presumption was that if we have two bacterial species that are really, really close to each other, evolutionary close, the 16S RNA genes would be very, very similar. Does that make sense? So basically, take all species of bacillus. They all have super similar 16S RNA genes. If you compare bacillus to, say, I don't know, staphylococci, there's difference. You compare staphylococci to Pseudomonas, there is difference. Makes sense, right? So he was studying bacteria and everything was perfect. He was categorizing. He was like librarian, okay? Putting bacteria in a certain boxes, certain drawers. One day he asked for a specimen from his neighbor down the hallway, and it was University of Chicago. Regular, like nothing special, one bacteria. And when he got the sequence of 16S RNA, he thought that there was a mistake. Of course, he didn't do the experiment. His grad students did. So he told them to go and repeat the experiment. It was really mean from him because the whole experiment lasts about a month. So they spent another month repeating the first one. Got the same result. And what Carl Woese realized is that he just stumbled upon a new domain of life. So Woese continued working, and now they generally accepted, all scientists accept this classification, that life can be first and foremost divided into 3 domains based on their genetic similarity. That's important. Genetic similarity. Those 3 domains are bacteria, archaea, that's what Woese stumbled on, okay, and eukarya. So those are 3 domains of life. What was shocking to everybody when data started to accumulate is that genetically archaea and eukarya were pretty close to each other. If you would look at the picture here, the phylogenetic tree of life, you will see that it has one original stem that's sometimes referred to as universal common ancestor. Okay, that's some cell that we all are descendants of. Okay, and then this universal common ancestor splits. One branch runs to become bacteria, and another branch runs to become archaea and eukarya. So essentially, in terms of like relatives, archaea and eukarya are basically brother and sister, and bacteria is the cousin. And this is something that scientists for a long time had to kind of, get at peace with, because they couldn't expect this, that archaea would be closer genetically to eukaryotes than bacteria. What about Whitaker? Did his classification become obsolete? No. As many things in science, it got incorporated into a new classification. So now we have 3 domains: bacteria, archaea, and eukarya. And inside of the domain eukarya, we have kingdoms. We have kingdom of protists, kingdom of fungi, kingdom of animals, and kingdom of plants. You see, that's what I really like about science. It rarely trashes something. It tries to incorporate the old discoveries that may not be 100% correct into a new development. We're good so far. Any questions before we move on? No? Okay. Now, a few things about cell theory and beginning of life, sort of. Well, first of all, just to make it clear, it's kind of not really, well, kind of microbiology question, but beginning of life is not a question of science. Science doesn't work with single events. Makes sense, right? We can't repeat origin, the start of life, so forget it. Scientists don't really care how life started. It's more important to figure out how it has evolved. But in about like 16th, 17th century, a lot of people thought that life can appear spontaneously. Like mice just appear in the house. Yeah, mice, roaches, for that matter, flies. Okay? Until brilliant Italian scientist Francesco Redi devised extremely simple and elegant experiment. So he got 3 jars with meat. Okay? One jar was left open. One jar was closed with a cork stopper. And one jar was covered with a cloth. And what he discovered, well, open jar, after some time, obviously, they're going to be maggots. The larvae, regular flies. Okay? In the jar that was protected, there were no maggots. So, apparently, life doesn't appear spontaneously. But interestingly, on the jar that was covered with cloth, maggots appeared. They appeared on the cloth. So, Redi basically demonstrated flies can produce larvae, maggots, okay? But meat is just basically sort of an attractant for flies. It's not something where flies are coming out or maggots are coming out. That was the first pretty significant blow to spontaneous generation theory. There were a lot of experiments afterwards, but the final and perhaps the most elegant knockout blow was delivered by Louis Pasteur in 1895. Those experiments were radically simple. Just genius simple. Pasteur had two flasks with a curved necks. Each flask contained a broth that could support growth of bacteria. He boiled them to kill all the living forms. And then he left one flask intact with a little bit of water that evaporated and condensated here, basically making a stopper. So there's no contact between the content of the flask and the air. In the second flask, he broke the neck. No surprise that this flask eventually got contaminated, simply by microorganisms getting there from regular air. This flask can be seen in Pasteur Institute in Paris, still sterile. 120 years, and a change. No growth whatsoever. That's pretty impressive. So, it was kind of not really an acceptable view anymore, spontaneous generation. So, on top of that, scientists like Schleiden, Schwann, Virchow, they discovered a cell. Now, it's always amazing if you think, the most trivial concepts, like cell. We all know that, they're cells. They were discovered in the 19th century. And it was a fundamental idea. Cell can only be derived from another cell. There's no way it can appear spontaneously. We don't know it. There was a foundation of the cell theory. Okay? And that was really, really crucial to show that cells, they constitute the fundamental unit of life. Okay? Now, you got to understand that, knowing that things consist of cells, knowing that these cells can cause disease, two different kinds of knowledge. The idea of contagious disease was known from ancient Greeks. They knew that some diseases can be transmitted from person to person through the air. Practical Romans actually built the first sewer system. They actually had toilet, not like running toilets like we have, but they had toilets that they weren't flushed. They basically just, all the sewage went directly into the river Tiber, which is next to Rome, okay? But they realized that they knew that sewage, human waste, can not only pollute the streets, it can contribute to the transmission of disease. So that was basically a public health measure, okay? But the link between microorganisms and disease was not established until 19th century. And there are 4 fundamental figures that I want to talk about, because some of them are really good stories. First was Ignaz Semmelweis. He was a doctor, kind of a hospital chief doctor, he was working in Vienna, being himself of Hungarian descent, and he was perplexed with the mortality and morbidity in the maternity ward. Women who gave birth, it's mid-19th century, often suffered from the condition called puerperal fever. You have not heard about it, I guess. That's great. Puerperal fever is the different name for sepsis. When a woman gives birth, there is a lot of blood involved, and any kind of infection will go directly into the bloodstream and can cause sepsis of a mom and can kill mom. Mortality rates in the maternity ward at some point reached as high as 30%. Imagine this, every third woman that gives birth dies. That's pretty terrible. So, of course, Semmelweis, as doctor, was not at peace with this. So he started to look for patterns. He started to look for the cause. Why does it happen? You noticed an interesting thing. That mortality, those deaths, occurred mainly in women that were cared by the medical residents. If delivery was performed by a midwife, there were much more chances that women will survive and will have no puerperal fever. And so he saw the definite pattern. Okay, residents contribute to the mortality more. Why? He quickly figured that there was one specific place in the hospital where residents went regularly and midwives went never. Any idea? For residents, it was a duty. They had to go there to keep studying the morgue. No gloves, no hand wash. They did autopsies and then came back to the maternity ward and delivered babies with those same hands. So what Semmelweis did, he introduced chlorine hand wash. That's it. Mortality fell to under 5%. It's all not rainbows and unicorns, because when Semmelweis delivered his findings at some of the conferences, his colleagues were vehemently opposing him. They said, no, that's all bullshit. He got ostracized, basically, got paranoid, didn't speak to anyone since then, and died in a mental asylum where he was placed for his paranoia by his wife and his best friend. So yeah, that's pretty sad story. And since then, Semmelweis syndrome refers to a situation when clear and obvious data for some reason are opposed by the community. Okay. Joseph Lister, on the other hand, had a pretty good career, unlike Semmelweis. Joseph Lister was a surgeon in Britain, and he developed carbolic acid wash. Carbolic acid is another name for a chemical known as phenol. It is a very aggressive chemical, but it's also a very good disinfectant. What Lister suggested is to spray carbolic acid on the surgical wounds to prevent post-surgical infections. Now it sounds absolutely barbarical. I don't know. Imagine somebody would say, oh, let's spray 70% rubbing alcohol on the, abdominal incision or open abdomen just to prevent an infection. We're going to do it every 15 minutes. Sounds pretty terrible. So carbolic acid spray was pretty terrible, but so was the surgery process anyway. If you would Google, that's actually the spray. Looks very steampunk, I think. That's the spray that they used during the procedure. That's actual Lister's spray. If you would Google Joseph Lister and look into the images, there's going to be one image of Lister performing surgery. And what you can notice is that there's a patient on the table, right? And there's one specific person that stands in the hand of the patient holding the hand. This time there was no anesthesia. But you know that properly secured patient doesn't require any anesthesia. So they secured them well. Actually, Lister provided consulting services during appendectomy of one of the European monarchs who got appendicitis right before the coronation. I don't remember the name. I think it was German. And the guy survived because majority of people died not of the surgery. They died of the infection after the surgery. So this guy didn't die, and he got Lister a ton of money. This, like I said, that I'm a king just because of that guy, because I didn't die. Okay. Pasteur later demonstrated convincingly that, say, rabies is caused by infectious agent. And finally, Robert Koch is considered often to be a founder of modern epidemiology. He developed so-called Koch's postulates that allow us to link an infection and the pathogen, the disease and the pathogen. It doesn't work that simple, but basically the idea is that if you have a bunch of sick folks, okay, you have healthy folks, you isolate microorganism only from the sequence, okay, then you infect some model, okay, and you see that this model shows the same symptoms, which is kind of hard, but let's say it does, okay. So you culture the agent. That's important, but becomes less and less important. But you isolate the agent, you grow it, you infect model organisms, and you look what happened to them. If they die, just like as original ones did, you're good. That's a causative agent. That makes sense? Last important step, you have to isolate that same microorganism from the dead model. Why is that important? You want to make sure that your model organism, your experiment, this model organism died from infection that you did, not something else. Does that make sense? Good? I kind of feel obliged to show this slide to you. Microbes are important, right? What for? Decomposition. If there would be no microbes, it would be quite literally neck deep in dead bodies. Food, microbiology, big deal. If we will start like bread, beer, wine, cheese, dairy products, all fermented products from sauerkraut to kimchi, microbes, a lot of chemistry. We will see that a lot of organic solvents produced by microorganisms. They're everywhere. In fact, there's probably only one kind of place on Earth where you're going to find no microbes. Volcanoes. Like inside of the volcano. It's too hot. Okay. Like these tar pits. So, La Brea, California. Molten tar. Scientists analyzed molten tar. There were microbes. In the molten tar. Living microbes. Which is pretty cool. Okay. But of course, we care about clinical importance of microorganisms. To give you a perspective, this is only for the United States. Okay? Mortality rate, meaning how many people die every year from a certain disease per 100,000 of population of the US. So we now have 300 million, so you can easily multiply this number by what, 3,000? Yeah, by 3,000, and you will get the total number of deaths. So every year, about 130,000 people die from infectious disease. In 2014, 51,000 Americans died of influenza or pneumonia. That's pretty considerable. I don't know why they grouped them together. Yeah.
Don't AIDS patients typically die from pneumonia?
Not anymore. We have drugs. They can die of pneumonia, mostly pneumonia. They can die of diarrhea. They can die of cancer. Pneumonia is probably the fastest killer. But if you would look at AIDS, they don't have any mortality data. What they do have, which is percentage change, you see this 85% change. It means that in the period from 1980 to 1995, the number of people that died of AIDS increased by 85%, which is a lot. And then since 1995 to 2014, the number of people that died of AIDS decreased by 10%. We have such good drugs against AIDS that it's considered a chronic disease. AIDS is basically same as diabetes or hypertension. With the proper care, the life expectancy is the same as for a non-infected person. We're going to talk about AIDS. Vector-borne, a lot of people die from infections with the drug-resistant bacteria. So if we multiply it by 3,000, 12,000 people a year, this is, I think, the most tragic statistic. We still have 2.4 thousand people that die from vaccine-preventable infections in the United States. Okay, something that can be taken care of, but isn't. Anyway, I think I got your attention to importance in the healthcare, and we'll talk more. I want to say about a few words for the good bacteria right there. Bacteria can deliver a bunch of solvents. Of course, you don't have to memorize all this metabolic pathways, but actually think about this. We now know how bacteria make those solvents. They can make ethanol. You all know ethanol. They can make acetone. You all know acetone, nail polish remover. They can make butanol. Now, butanol is a very promising future fuel, alternative fuel. What you can do, you can pour butanol in the gas tank of your car instead of gasoline. That works just the same. And you can make butanol by fermenting, say, corn. Okay. The only problem is that the bacteria called Clostridium acetobutylicum, the yields are terrible. The highest yield that they could make is 2%. So you get in the solution, like in the final broth, you have 2% of butanol. It's really low. They try to make it better. In France, in California, studies of microbiome changes in the wine during fermentation. So scientists look at changes in microbial populations in the wine as it becomes wine. And that's crucial because think about this. If you know which composition of microbiome delivers you the best wine, you can make that starter culture. And all you need to do is to add grapes, and the starter culture, the mix of bacteria, will deliver you the best wine. And this was actually shown in France, that why wines from different regions have different taste. Moreover, why wines from, I don't know, southern slope may have different taste from the northern slope of the hill. The reason is that microbes that are on the grapes are different. Different fungi, different bacteria, different populations, different proportions. It's all really important. So that's a really big story. And last but not least, new antibiotics. We're losing wars. Last year, United Nations announced that we officially lost the war on drugs. To drugs. In medical field, we keep losing the war to antibiotic resistance. More and more microorganisms become resistant to antibiotics that we have. So developing new drugs becomes more and more important. And other microorganisms can help us with that, for instance, and sources can be absolutely bizarre. Bacteria in the ocean, isolated from the ocean, can deliver new promising antibiotics. Now, this is something like we're going to make certain points throughout the course. And one point is, did you ask yourself, why do bacteria or fungi produce antibiotics, chemicals that kill bacteria or fungi? What do they use those chemicals for? Huh?
To defend themselves.
So again?
To defend themselves.
Against?
Other bacteria.
Perfect. Yeah, it's a chemical weapon. Yeah. They produce antibiotics to kill other bacteria, to kill competitors. Okay? And that's the whole idea. And obviously, say if Saccharomyces spora produces antibiotic called daptomycin or taromycin, okay, then Saccharomyces spora is not going to be killed by that antibiotic. They produce poison that they are sort of immune to. Does that make sense? So this is a point that I want to make like early in the course, and it will be useful for you when you work in the clinical settings. Resistance in bacteria can be induced by humans. We contribute a lot. But you have to understand that there are microorganisms that are naturally resistant, normally resistant. There are bacteria that are normally resistant to, I don't know, neomycin that you can find in an antibiotic cream, or bacteria that are resistant to tetracyclines. It's normal. Not all of them, but some of them, they are. That make sense? So we're going to take a break, okay?